All right, so I suggest to move to the next presenter. Again, thank you very much, Wei Shang, always very eloquent and clear. Our next presenter of the day is Jess Cardin. Right, Jess? I don't think I ever met you in person, actually. You worked together with Mike. We had a few interaction with Mike Higley in the Higley Cardin lab. So Jess is an associate professor at the Kavli Institute for Neuroscience in the Yale Department of Neuroscience. Thank you very much for joining us, and again for all the work that you are doing together with Mike. So while we know that the RAI is the central factor in SMS, we don't know how its disruption alters the development and function in the brain. So the Higley Cardin lab at Yale combines a microscope with genetically encoded sensors that light up in the brain of mice when neurons are active, and providing a readout of brain function with extremely high spatial and temporal resolution. It's actually quite impressive to see the data. With these tools, we can investigate how patterns of a neuronal signal works. Now, by imaging neurocortical signal in mice models of disrupted RAI1, we hope to identify unknown functional signatures of the brain activity associated with SMS. And with that, I would like to let Jess start her presentation.
So Carol, you'll make her a host, please.
Jessica, I just made you a host. If you wouldn't mind, please hitting record. I'll get the recording from you later.
Sounds good. Let me find the record button.
It's below, and it's a red button.
Yep, got it.
Thank you very much.
Okay. So first, I really want to say thank you so much. It's really a privilege to be able to share some of the data from this project with you all. This is a project that is entirely supported by the SMS Research Foundation and would not have been possible at all without that support. And this particular project is a collaboration between my lab and Mike Higley's lab at Yale. And I'm going to tell you a little bit about the methods that we're using, and then I'll show you a little bit of what that data looks like and some of our most recent results. So we're really interested, as Gianluca said, in phenotyping alterations in brain activity in mouse models of SMS. And I think everyone here is really invested in identifying the relationship between dysfunction of the gene RAI1 and SMS. And looking at the brain, there are a lot of levels to unpack in between the gene and the phenotypes that we see in the syndrome. And that goes all the way from signaling within individual neurons in the brain to the way that neurons talk to each other through synaptic connections, the activity profile of individual neurons, and then the activity of populations of neurons as they're talking to each other during cognition and behavior, how those circuits can be organized, and in particular, the dynamics of brain activity in large networks. And one of the reasons that we're really quite focused on this level here that I'm highlighting, the network dynamics, is that we know that brain functions like learning and memory, perception, cognition, language, these all are functions of large-scale networks of interaction among different brain areas, particularly in the cerebral cortex, which is really important for cognition, for language, for perception, and those kinds of functions. So we'd really like to focus in on how RAI1 dysfunction disrupts network dynamics in the brain. Our model for this is, as in the previous talk, is a mouse model in which we are disrupting the function of either one or both copies of RAI1 using an inducible Cre system. So this is a mutation in specific populations of neurons. In this case, it's all of the excitatory principal neurons. So about 80% of the neurons across the cerebral cortex. And we are introducing an indicator to report neural activity using a method that we developed a few years ago, where we can use adeno-associated viruses to infect the whole brain all at once to express GCAMP6, which is a calcium indicator. And the reason that you would want to put a calcium indicator into the brain is that calcium is a really excellent proxy for the activity of neurons. And this allows us to read out in fluorescence how active neurons are across the brain. But we don't really, in this case, want to look at just single neurons because we really want those whole networks that span multiple areas of the brain. So we really want to zoom out and take snapshots of as much of the, in this case, the cerebral cortex as we can capture at once. So in this case, we're taking snapshots in real time using something called wide field or mesoscopic imaging. We're putting mice underneath a microscope. Where we have an LED to illuminate the top of the brain with light, and then we have a camera to capture fluorescence. And this is what that looks like. So this is a top-down view of a mouse's brain. This is sped up a little bit, so it's not in real time. And what you're seeing is fluorescence that indicates the activity of populations of neurons. So warmer colors indicate more brain activity. And just to orient you, these are the two sides of the mouse's brain. This is the front and this is the back. And so the nose would be up here and the tail would be down there. And the cool thing about this is that it's pretty non-invasive. We get these snapshots right through the skull of the mouse. Okay, so we have two goals with this kind of project, right? One is that we really like to understand the links between RAI1's function as a gene, the patterns of brain activity that correspond to cognition or behavior or different states of the animal. Right, and how that relates to some of the behavioral phenotypes that we see in SMS. But as a second stage, one of our other goals is really to develop a platform that works as a sort of moderate throughput screen. We don't just want to understand what's disrupted about brain activity. We want a way to screen for the impact of potential therapeutic strategies to rescue perturbed brain activity. And those could be gene therapy, or they could be small molecule or pharmacological manipulations. But we really want something that gives us clear phenotypes of brain activity, clear benchmarks for potential therapeutics, and we want it to be rapid. We don't want this to take years. We want to have a really, quick assay for rescue of perturbed brain activity. And one of the nice things about this technique is that it gives us rapid snapshots. It only takes about half an hour to an hour of data per mouse to get phenotypic snapshots for each animal. We can do this imaging throughout the lifetime of the animal, from development all the way through adulthood. We can do within animal comparisons across a pretty long time scale. Okay, so to give you some examples of what this looks like, so on the left-hand side, I'm going to show you data from a wild-type animal, which has 2 functional copies of RAI1. And on the right, I'm going to show you a movie of brain activity from a homozygous mutant mouse in which both copies of RAI1 have been lost. So on the left-hand side, tails on the bottom, and the left and right hemispheres of the brain, you're seeing a top-down view through the skull. And in wild-type mice, you see big patterns of activity. Again, warmer colors mean more brain activity. So this is a mouse that's sort of hanging out, going through lots of different behavioral states during wakefulness. You see lots of big patterns of activity on both sides of the brain, as well as pauses in between those big bursts of activity. And waves of activity will move between brain areas. Now, if we then compare to our homozygous mutant on the right-hand side, if you watch the activity here, you'll notice a couple of things look a little bit different. We do see some large bursts of activity, but far fewer than in the wild-type mouse on the left-hand side. And a lot of the activity that we see appears to be more diffuse and at a lower level. So here we see less of those bright yellows because the activity is a little lower. And much of the activity appears to be a little more homogeneous across brain areas as opposed to punctate and moving in discrete waves. So this kind of snapshot gives us some clues to what to look for in examining patterns of brain activity in our different mutants. So I will take you through some of the ways that we analyze this kind of data in our awake behaving mice. One of the cool things about mice is that they do a lot of the same things that humans do. So everyone who's listening to this presumably has noticed at some point that, sometimes you feel like your brain works differently at different points in the day. You have moments where you're really alert and focused and attentive, and you feel like you're able to process information quickly, form memories, retrieve information. You're really on. And then you have other moments where maybe you're a little drowsy or a little sleepy, or you're tired, you're worn out, and you're not really processing information as well. So mice also similarly go through different states of either deep quiescence and drowsiness, or moderate arousal, or very active moments where they might perform a motor action like grooming or running. So what we do is we divide our data into different behavioral states to look and see what the relationship is between activity in the brain and the different behaviors that the mouse is exhibiting. And here I'm showing you 3 different categories. One of the ways in mice and in humans actually that we categorize arousal or active states is to look at facial movements. So here I'm showing you on the left-hand column a high face state, and this is the mouse sitting still, not doing any overt motor movements, but showing lots of motor movements in his face. Humans do this too when we're alert and attentive. So on the left-hand side is a moderate arousal state of high facial movement. In the middle is grooming, which is a repetitive motor activity. And on the right-hand side is a very high arousal state where mice start to run on a running wheel. And our wild-type mice are on the top. So these are regular mice with 2 functional copies of RAI1. In our middle row are our heterozygous mutants, which are missing 1 copy. So they have 1 functional copy of RAI1 left. And on our bottom row, homozygous mutants, which have no functional copies of RAI1 left. And immediately you can see that the difference between our wild-type mice and our mutants is that our mutants, over all these different behavioral states and different behaviors that they produce, have a less activity in the cerebral cortex in this top-down view. And again, warmer colors would be spots with more neural activity, more firing of neurons. What's interesting about this data is that we appear to also have a bit of a dose-dependent effect, where our heterozygous mutants have a modest reduction in overall activity levels, but our homozygous mutants have a more substantial reduction. Importantly, this isn't because the mice are exhibiting different behaviors. In this plot on the upper right, you can see our 3 categories: wild types, heterozygotes, and homozygous mutants. All 3 groups of mice exhibit the same behaviors in about the same proportion. So there are no major differences in the behaviors of these mice. They all exhibit these 3 different behavioral states at about the same rate. And if we look a little more quantitatively at activity levels across the brain, we find a sort of stepwise function where our wild-type mice in blue show the most neural activity, our heterozygotes in orange show less, and our homozygous mutants with no functional copies of RAI1 show the least activity. And this is true across the brain. So here I'm showing you an area in the back of the brain. This is visual cortex. An area in the middle of the brain. This is somatosensory cortex. And an area in the front of the brain in motor cortex. And across this front to back axis, we see an overall decrease in neural activity in the cortex in our RAI1 mutants. Okay, so there's one phenotype here easily seen in this kind of data. Now, the other thing that we can get from this kind of snapshot of neural activity is something called functional connectivity. And you may have heard of this in people discussing MRI data from humans. Functional connectivity is just a correlation-based measure of the relationship between activity in different parts of the brain. So how coordinated is neuronal activity in 2 different parts of the brain at any given moment? And again, I've divided data here into 4 different behavioral states with increasing arousal as you go to the right. And again, our wild types are on the top, our heterozygotes are in the middle, and our homozygous mutation mice are on the bottom. In each case, I'm showing you a matrix of correlation values where all the different areas of the brain are shown on each axis. And so the values that you see here are color code indicating how correlated are the patterns of activity in 2 different brain areas. Now, these plots are, I admit, kind of hard to look at. There's a lot of yellow and green and blue here. So in a moment, I'm going to show you something that's a little easier to interpret as you look at it. But just by eyeballing this, you can sort of see that there's more warm colors, higher correlations in our mutant animals. Now, if we take this kind of plot and instead calculate the difference between our control or wild type animals and our mutants, a pattern starts to emerge. So here I'm showing you the difference in the degree of functional connectivity or correlation that's shown by our heterozygous mutants on the top and our homozygous mutants on the bottom. So warmer colors here indicate enhanced functional connectivity or correlation. Cooler colors indicate a decrease in functional connectivity. Overall, across all the different behaviors that we've seen, all the different states of the animals, and in both heterozygous and homozygous mutants, we see an overall enhancement of functional connectivity. So activity is more coordinated across the cortex in both sets of mutants here. I'm suggesting that although there's decreased total activity seen in these animals, the coordination of activity across areas has been greatly increased. So this is sort of the end of our first pass at looking at these kinds of experiments in these animals. And what we've come to is two clear phenotypes in the neural data from the in vivo imaging in these animals, a decrease in overall neural activity and amplitude, and an enhancement of cross cortex functional connectivity in the mutants across multiple different behavioral states. So now we can start to think about going back into this kind of screen of neural activity to compare the activity in our mutants to that in perhaps mutants treated with either gene therapy approaches, small molecule approaches, to see whether treating animals perhaps at different age points, different points during development, gives us different outcomes in terms of amelioration or rescue of our neural phenotypes. So all the data that I just showed you was collected by a very talented graduate student named Rachel Oren. This is her main project in the two labs. She's co-mentored by myself and Mike Higley. And thank you very much for listening.
Excellent. Thank you very much, Jess. A round of applause virtual. Thank you for your talk. Again, for anybody, any question you may have, go to the Q&A function. As we wait, I'll start with one question. Just to know that the RAI is known to have a role also in the sleeping cycles and the activity during sleep. So is it possible to do this type of measurement as mice are actually sleeping? I don't even know if it's technically possible, but I'm wondering if that could be done and that could show if there's truly a difference in activity during sleep.
So we, in this initial project, we really focused on developing a reasonably high throughput screen. So we really wanted that fast screen, like, look at every animal for a half an hour or an hour to really process through looking at comparative approaches. But at the same time, we've been developing a parallel set of technology with a head-mounted version of this microscope. So we now have a freely moving version. So we are able to do sort of full circadian imaging in mice freely moving in home environments. So our hope is that we can also catch sleep-wake transitions and activity during sleep in the same animals. That's not a high throughput screen in the same way, but certainly our faster high throughput screen has pointed us in some really clear directions that are worth digging into with a sort of more long-term approach. Absolutely.
Wow, very fascinating. Thank you. So look forward to seeing some of that. And and we heard before about the BDNF activator. I think that's certainly the small molecule that we could test in your system. Maybe a collaboration between you and Professor Wei Zhang could be a way to go here to accelerate some of our research.